All right, today we're going to wrap up chapter 16. So just kind of as a overall making sure that we're comfortable with where we're at from what we talked about before. We left off with Watson and Crick discovering, and well, Mendelssohn and Stahl too, discovering kind of how DNA replicates in just a really general way. And so we talked about how we discovered the semi-conservative model is what won out, where we take an existing double helix, so two strands of DNA, we split them down the hydrogen bonds that are in the middle, and we use each half as a template to rebuild the other half, which works out well due to this complementary base pairing where adenine only wants to stably chemically adhere to thymine, and cytosine only wants to stably chemically adhere to guanine. And so that meant that all the other possible methods, the conservative one and the dispersive one, were ultimately crap. So we discarded those. And so that's what we're going to kind of pick up with, is we're going to start talking about exactly how this process of splitting the existing strand to form two new strands will work. And so we're going to do this from a very molecular standpoint, where we need to make sure you guys know the chemicals involved in making this happen, because this will be a little bit more of a symphony versus just, one magical thing suddenly occurring. So DNA replication is going to start at something called the origin of replication. Origin means start, replication means replication. You'll also see sometimes they'll refer to this as the replication fork as it continues. In prokaryotes, you oftentimes will just have one origin of replication, whereas in eukaryotes, to speed this process up, we oftentimes will have many. So while origin means, the start, if you will, there can be multiple origins that occur on one piece of DNA. Now, this replication fork is where we're actually splitting it, and so this will kind of move along as we unzip the strand of DNA. Now, there'll be two guys that are involved with that. The first guy is going to be topoisomerase, which kind of straightens out the DNA to allow us to more easily split it. And then helicase follows him, and helicase acts like an unzipper in this case, where it unzips the DNA by breaking the hydrogen bonds and splitting the previously single double helix into now these two separate strands that are missing their partner. And so each of these strands then will be acted upon by a series of other structures. You'll see these guys here called single-stranded binding proteins, and their job is to basically just block things so that the DNA can't come back together. You don't want the DNA to go, hey, you're the opposite of me, we're complementary, let's get back together. You don't want to reunite the band, if you will. And so you need to have these little single-stranded binding proteins, the Yoko Onos of the molecular world, and they will prevent anybody from getting back together. So that allows for us to treat each template separately. We'll then have primase, DNA primase. Wait a second, piece of crap. RNA primase. This is wrong. And RNA primase is ultimately going to attach a small sequence of RNA nucleotides to the DNA as a marker, because we need to know where to have polymerase, because DNA polymerase is the guy that's going to be coming up soon. We need to make sure we know where to have him bind. And so in this case, we're going to have, after this primer is put in, these few RNA guys. You can see they're trying to show it by listing these little brown pieces there to show that they're not DNA nucleotides. You'll use those to allow DNA polymerase to come bind and to start building the strand. And so it will always build in the 5' to 3' direction, always. And so it's always going to be building towards 3', if you will. And that will be on the strand that's being built. So this strand, which you can see bends up here, is being built towards the 3'. It's being built this way. That means this strand, which will always be the opposite direction, has to be built in the opposite direction. And so that means this strand has to be built this way. So one strand is going to be being built towards the replication fork, and one strand is going to be being built away from the replication fork. And this will create some problems for us later that we'll talk about. And then lastly, after DNA polymerase has come in and binded all of our proper DNA nucleotides, DNA polymerase will come back through, remove and fill in these primers of RNA with DNA, and DNA ligase will come through following DNA polymerase and stitch together all the gaps. So what that means is I might have where there's a bunch of DNA nucleotides stuck together like that, but then I had to come back and patch that primer. and then maybe there's another section where it's a nice long strain. But when I patch it, it's not connected. There's the backbone has these little splits. And so ligase comes in and just polishes those guys off to make sure it's one complete, nice, neat section. So those are some of the major guys: topoisomerase to straighten, helicase to split, single-stranded binding proteins to make sure it doesn't come back together, and then RNA primase initiates things with a primer, lets things know where to start. DNA polymerase will go through and rebuild the strand and replace the primers, and ligase comes at the end as a patching crew to make sure everything's running nice and smoothly. So a couple things to make sure we're okay with. Antiparallel just refers to the fact that one template strand is going to be from 5 to 3, if you will, 5' to 3'. That just refers to which guy is sticking off at the end. Is it a phosphate or a sugar? So the sugar end is the 3' end, and the phosphate end is the 5' end. And then the other guy has to be the opposite, because just like a road, we've talked about this, I believe, but just like a road, they face opposite directions. And so if you look down here, this one ended in 5'. Its previous partner, this is the other template, ended in 3'. So what this means is that strands have to be built in opposite directions. Now the guy that's being built towards the replication fork, that's called the leading strand, and that's the guy that has it easy, because it needs to keep adding bases, but you keep opening things up, so there's always more bases to add. So it's kind of like, imagine anytime you're about to run out of something, someone's sliding you more. So I don't have to worry about running out. I just keep going. I don't have to stop and change or anything else. So the leading strand is built essentially continuously, is the word I would normally use here. Whereas the lagging strand has to be built away from this replication fork. So that means if I go ahead and attach my primer and start to build, I'm not going to make it that far before I bump into kind of what I've already built. And while I'm building this section, this is unzipping, but it's unzipping in the other direction. So it's like you're driving your car away from something, but the road's being built in the opposite direction. And so if you want to go drive on the new section of road, you have to detach and then go reattach where the new section is and start driving away from where they're building the road again. And so we end up having to build these in a series of fragments, which we call Okazaki fragments. And so these Okazaki fragments will just be short segments of DNA that we're going to build because we have to keep going back and starting at the replication fork, but the replication fork keeps moving further and further back. So every time we kind of build it and we're like, yeah, we got it all done, more have opened up behind us, so we have to go back and start another section. And this keeps going and going until eventually you have completely split the strands and you're done. And so this type of building is going to require a lot more of DNA ligase, because while you build it similar, you're going to have a lot more primers, you're going to have a lot more sections built, because we have to do it in so many pieces. And so ligase just has to come in and make sure it fixes all those pieces together so everything's okay. When you talk about the leading strand, you do need ligase to patch up the primer and such, but for the most part, the leading strand is really a primer piece that has to get replaced and fixed, and then the main piece. That's it. You know, there's not a whole lot of pieces there. And so you don't need a whole lot of ligase in terms of function to make the leading strand work, because the leading strand is just easier. So that describes antiparallel, and that lets us know what the leading strand is versus the lagging strand. And you should remember, antiparallel is the reason that we have these, because we can only build things, polymerase can only build things in a 5 to 3' direction. That's our problem. If polymerase could build stuff in both directions, this wouldn't be an issue, because it could build one strand from 5 to 3 and the other strand from 3 to 5. The other piece I want to address is the template strand here. If we look at the top, is 3' to 5'. So if you look at that, it's easy to think that ultimately you would be building this new strand towards the replication fork and that it would be the leading strand. But in reality, they're antiparallel. So that means if this strand is 3' to 5', the strand that we're building has to be the opposite. So in other words, it has to be 5' near the replication fork and 3' away from the replication fork. And so this strand has to be built away from the replication fork, so it's actually the lagging strand. So same thing down here. It looks like it's going to be built 5' to 3' away from the replication fork, but because the new strand is the opposite, it's actually being built 5' towards 3' at the replication fork. So in other words, this looks superficially, if you look at the template, as though it's the lagging strand. But when you actually realize that it's anti-parallel, the new strand is going to be the leading strand. So just make sure you remember that, because if I put it on something, I don't want you guys to freak out and get it wrong because you just didn't quite pay attention. Now, the last part of this section really focuses on fixing DNA if we manage to screw something up. So if we manage to screw something up and it's from replication, but we don't catch it right away, we normally do a mismatch repair. So this is kind of internal issues where we screwed up while we were doing replication. Excision repair normally deals with things where like UV light or carcinogens or something else external has jacked up our DNA, but it had nothing to do with replication. So both of these are going to try to fix errors in the DNA. It's just how you got the error is what kind of deals with whether it's a mismatch repair or excision repair. But the general idea is that if you can see something's wrong, and DNA polymerase and there's other enzymes that can go through and kind of proofread. So if they see something is wrong, they'll oftentimes send an endonuclease, or sometimes just called the nuclease, but endonuclease to the site, and the endonuclease will essentially cut out a chunk of the DNA. So it'll take one side where the error is, and it'll just kind of scoop out a little section. And then afterwards, you can have DNA polymerase and DNA ligase can come in, and they will just rebuild that small section, and DNA ligase will attach it to make sure it's all smooth and good and everything's fine. And so aside from just having where this process of replicating DNA is very accurate, you're talking about 1 in 10,000 nucleotides that you might see an error. But then beyond that, you start to proofread it, and so now you start pushing the errors into more like the millions. And then when you go through and you continue to do proofreading and such, you get to where there's going to be very, very, very few mistakes. I don't remember the exact number, but I know it's well into the millions, if not billions, before they tend to make a mistake. Now, that can still be significant, because when you look at humans, I believe we have about 5 billion nucleotides, and if I remember right, and I could be wrong, you have about 1 mutation every 1 billion nucleotides. So for a human, when we do replication, we might get 5 mistakes. Now, lucky for us, most of our DNA is not coding for a protein. You know, there's lots of sequences that won't matter that much if they get screwed up just a little bit, and some mutations don't matter anyway. And so it's not that big of a deal. But when you think about if I gave you a test that had 5 billion questions, that's asking quite a bit if I say you're allowed to make 5 errors, even if that's what the situation is. So this is a very accurate process where it makes very few mistakes, but even if it does, it can come through with endonuclease to cut it up, polymerase and then ligase to patch it up, so we're back to normal. There's no long-term damage that we have to sustain. And so this can help minimize our risk of cancer. You know, this can help minimize our risk of just having, in general, faulty genes, regardless of whether they're going to harm us in our everyday life. And then telomeres. This one's used largely for our cells to figure out how old they are. So when cells go through a process of division, there's ends of their chromosomes called telomeres, that just basically means end. And so these telomeres, every time you divide, just think of it like you take the first primer that's at the very end, and you pull it off and you just leave it. You don't replace it. And so that end shrinks just a little bit. And so as a cell divides over and over again, the ends, these telomeres, can shrink down. And so the size of the telomere can be used as a gauge for essentially how old the cell is. And by how old it is, we mean how many times it's gone through the cell cycle, cell division. And so an old cell then would be one that's done a lot of cell division. And there is an enzyme that can replace this. So when you're going to give birth to a child, you don't want to give birth to some like the movies or something where it's like the little kid in the carriage with a cigar that's like a 40-year-old guy. You don't want that. And so ultimately, we have telomerase, and some organisms like lobsters produce this naturally, and so they will replace these telomeres, and so that way the cells don't really age, because the telomeres kind of fix that, just like we do with our young, and some things like tortoises and lobsters appear to do naturally, which is why they have negligible senescence, which means they really just don't get old. They can get older, you know, but it's not like they just die of old age in the way that we tend to. They normally die from something else like disease or predation. It doesn't seem like their body just collapses on them like ours does. But this is the method by which our cells determine this. And so oftentimes if a cell gets old enough, if it kind of does things where it's messed up enough, it seems like it's just aged too much, it can actually go through apoptosis, or it can slow down its metabolism, and it'll start to divide less and less. So sometimes when you see older people, you can like look at Mr. Coffin if you'd like, and you'll see that they tend to have like thinner skin, and you'll see like those spots on them because they have a lot of older cells because they're not replacing them as often. You know, everything's slowing down because it's like reading in its own telomeres that it's old. And so this is just kind of a cool thing scientifically, that if we can kind of figure out how to mess with this, but we don't want to tinker with it too much, because you can also have cells that kind of ignore things properly, where like cancer cells or something that don't go through and process these telomeres correctly. So we don't want to just go through and like take a bunch of telomerase and become Cancer Man. And so you got to be very careful, and we're trying to study other organisms and figure out how it is that they can use this telomerase, and they can essentially keep their cells being young indefinitely without getting some of these other diseases, disorders, mishaps that we sometimes associate with having improper cell growth, with having cells think that they're too young and get a little bit too eager about going through cell division. So telomeres are kind of a cool thing scientifically. If nothing else, then they drive home that age-old idea of mortality due to age. And so that's kind of a neat one. All right, so real quick review as we wrap things up. So enzymes, helicase, and you can actually add topoisomerase if you want there, and primase, these guys are all like the early guys, where they're going to split things apart and get them ready so we can actually build a new strand. So we're going to straighten it, split it, and prime it, because without those things we can't have the new strand be built. Then polymerase and ligase come in, and their basic tasks are to build and connect all those pieces so we end up with one coherent, nice, full, unabridged DNA double helix. You know, if we only had polymerase, we'd have all the proper nucleotides, but they wouldn't be fastened properly. We'd have breaks in the backbone. So these guys make sure it's all set. Single strand binding proteins just keep this apart so the DNA doesn't come right back together, so it buys us time so we can make each template find its proper nucleotide complements to rebuild each strand. And then we've talked about at the end here, nuclease is the guy that cuts out a section, or endonuclease as it's commonly referred to as, will cut out a section if there's a screw up. And then telomerase is the enzyme that will go through and patch these ends that kind of shrink, and the ends of the chromosomes that we use to try to figure out just exactly how old our cell is in terms of cell divisions. I hope you guys have liked Chapter 16. I'll see you soon.